Today, I will give you a talk titled The Dual Approach Strategy to Optimize the Safety and Efficacy of Pharmacopuncture. Complementary and alternative medicine can be used for the prevention of health program, health promotion, health maintenance, or other reasons. It becomes in a broad range of forms. Here, we will be looking at the most frequently used complementary and alternative medicine. Here you can see 5 types of complementary and alternative medicine listed. Many proponents of complementary and alternative medicine use therapy and healing practice from alternative medical systems such as homeopathic and naturopathic medicine. Alternative medical systems include also traditional medical systems from other countries, such as Indian Ayurveda and traditional Chinese medicine. Especially acupuncture and herbal medicines are categorized within traditional Chinese medicine. These therapies have long been practiced worldwide. Let me introduce here briefly the difference of acupuncture and pharmacopuncture. Acupuncture is a treatment procedure where needles are inserted at acupoint under the skin. Acupuncture have been mainly used in and around East Asia for 2,000 years plus. Based on its long history and the widespread use of acupuncture, various acupuncture techniques have been developed. Pharmacopuncture is one of the new acupuncture techniques. This combines two most frequently used traditional medicine treatment forms, which are acupuncture and herbal medicine. Herbal medicines are directly administered to acupuncture point for mechanical and chemical stimulation, which can maximize acupuncture point access and treatment efficacy. Acupuncture treatment has several advantages. The effect of pharmacopuncture was immediately observed after the treatment because herbal extract and animal product are directly absorbed without a need of passing with the gastrointestinal tract. And additionally, the patient with a difficulty on swallowing and those who refuse to take medicine or an emergency patient may profit by receiving pharmacopuncture treatment. Therefore, the major advantages of pharmacopuncture treatment are seeing a quick result with the additional synergistic effect of herbal extract and acupuncture and easily controlling treatment dosage. In this presentation, I will be addressing 3 subjects. First, current trend of pharmacopuncture use in hospital and clinic patient. And second, the range and frequency of heart issues and the significant adverse event in use of pharmacopuncture treatment compared to acupuncture. And third, standardization strategy in manufacturing process to improve the quality of pharmacopuncture, which is safe, stable, and effective enough. First, I will quick go over trend of pharmacopuncture use. In reality, pharmacopuncture treatment has been widely used in East Asia, including China and Korea, for thousands of years. Now it is rapidly growing in West also. In Europe, Biscum album pharmacopuncture. In Europe, the whole plant extract of Biscum album, known as European mistletoe, has been used as adjuvant cancer therapy and reduce of blood pressures. Even though most of its usage and the relevant studies are limited, in certain local area, the result of pharmacopuncture treatment are being accumulated, and so far it looks promising. In Russia, China, and other Asia, cerebral ricin derived from pig's brain tissues, which has potential neuroprotective and neurotropic properties, is used in the treatment of acute ischemic stroke. In China, over 200 herbal preparation types for injection are produced by pharmaceutical company. And pharmacopuncture is covered by some private insurance, including traffic accident insurance in Korea. Pharmacopuncture was introduced to Korea in the 1960s, and the use has become widespread since the 1990s. And the pharmacopuncture treatment in Korea has been developed with a decent clinical outcomes and the supportive experimental evidence out there. Pharmacopuncture treatment can be classified into 5 groups: animal-based pharmacopuncture, meridian field, 8 principle, single compound, and formula pharmacopuncture. Unlike in vivo and in vitro experimental studies, animal-based pharmacopuncture and meridian field pharmacopuncture were the most studied pharmacopuncture in clinical study. And bivenom pharmacopuncture placed first as among its pharmacopuncture as the most frequently used pharmacopuncture, followed by placenta, sweet bivenom, anti-inflammatory, 황연해독탕, and neutral natured blood stasis pharmacopuncture, etc. Pharmacopuncture treatment is mostly applied for the purpose of maintenance of immune balance and physiological homeostasis. And the major disease where this pharmacopuncture is used is musculoskeletal disorders. And it can be used also for treatment of other diseases, for example, cancer, obesity, digestive tract, urinary tract, blood circulation, or nervous system related disease. Next, I'm going to explain about adverse event of pharmacopuncture. Although pharmacopuncture treatment has many advantages, the increase in the individual case of adverse event and the decrease in the number of approved new drug applications have been steadily appeared. Adverse events mean a diversity of unexpected medical problems which could happen during acupoint injection therapy. Main adverse event associated with acupuncture or pharmacopuncture were presyncope episode, infection, pneumothorax, or allergic reaction. Of these, the most frequently reported adverse event was allergic reaction, which is the hypersensitivity to B-venom pharmacopuncture. Normally, B-venom is safe for most people when B-venom is injected under the skin. Some people might get redness or swelling where injection is given. Adverse event symptoms include associated with B-venom pharmacopuncture include itching, nausea, anxiety, trouble breathing, chest tightness, confusion, and low blood pressures. Recently, clinical trials on B-venom pharmacopuncture resulted in serious risk. The most severe adverse event was anaphylactic shock, which is potentially life-threatening whole-body allergic reaction to a chemical. The instance of this anaphylaxis was rare at 0.019%. Although this severe adverse event it's not common, and nor did they live sequelae. There's need for caution about that. And next, I'm going to talk about standardization of pharmacopuncture preparation. Medicinal materials used for pharmacopuncture treatment are produced by each different method of operator at discretion, which has a higher chance to cause adverse event than other treatment. Considering these problems, it's required to standardize the raw medicinal materials and the manufacturing process, and to confirm its safety, stability, and efficacy for production of high-quality pharmacopuncture. In this section, we will be looking at briefly the standardizing process to produce a high-quality bee venom pharmacopuncture by enriching effective component and by removing harmful factors. Bee venom is extracted from honeybees. It is bitter and colorless liquid. In Korea, this bee venom pharmacopuncture is usually used to relieve the pain and to treat several diseases such as cancer, arthritis, rheumatism, rhinitis, asthma, and skin disease. Several compounds in bee venom are known to exert their pharmacological effect individually or interactively depending on their concentration. The major compound in bee venom is melittin, which is a peptide showing anti-inflammatory, anti-tumor, and antimicrobial effect. Bee venom contains also the protein phospholipase A2 and apamine, which are allergens, as they are capable of inducing allergic reaction in susceptible individuals. Therefore, we have tried to establish the appropriate purification condition for the complete removal of allergens and the high yield of melittin from crude bee venom, resulting in high-quality bee venom pharmacopuncture. First, we have evaluated whether solvent itself influences the melittin content. Among various solvents we have tested, the aqueous ethanol was found not to affect to melittin content during purification and analysis of crude bee venom. To isolate and purify the active compound, the gradual change of element strength led to partition into several fractions. As you can see here, fraction number 1 has null compound. And in fraction number 3, apamine was detected. And in fraction number 4, both apamine and phospholipase A2 were detected. The melittin was eluted in 50% and 60% ethanol solvent. However, this eluent was contaminated with phospholipase A2. The melittin was neatly eluted in 70% and 80% ethanol layer with no apamine and no phospholipase A2. Therefore, the 70% and 80% ethanol solvent was found to be the best solvent for purification of melittin from crude bee venom. Increasing the conformational stability of protein is an important goal for basic research and industrial applications. Lot-to-lot data is also important to ascertain the stability of manufactured product. And that's why we first of all evaluate the change in melittin content per product lot. As you can see here, in first table, similar melittin amount was found in different lots. And then the stability of pure melittin was investigated on different conditions, which are pH, salinity, storage period, and temperature. As shown in this result, the groups which were treated with pH, salinity, and storage period appeared low levels of melatonin content compared to non-treated groups, indicating that change in pH, salinity, long storage period, and high temperature induced the destabilization of melatonin. Current available literature involving in vivo and in vitro studies suggest melatonin induces signal transduction leading to multiple cancer death mechanisms, including inhibition of angiogenesis, cancer cell proliferation and metastasis, and induction of cancer cell apoptosis. We found the newly purified B-venom pharmacopuncture induced apoptosis in human hepatic cancer cells in a dose-dependent manner. In addition, melatonin is known to have protective actions against several inflammatory diseases such as skin inflammation, atherosclerosis, liver inflammation, neuroinflammation, and arthritis. Melatonin inhibited the inflammation by suppressing the LPS-induced nitric oxide production and the gene and protein expression of inducible nitric oxide and cyclooxygenase 2 in macrophages. Regarding adverse event of B-venom, we have investigated the effect of purified B-venom pharmacopuncture on the regulation of inflammatory mediated production by mast cells, resulting in allergic immune response. As expected, melatonin protected the human mast cells against 48-8 induced granulation and histamine release and production of TNF-alpha and IL-4. Among various toxins, heavy metals and endotoxins are critical parameters for establishing safety of the medicine. We found the average concentration of 4 heavy metals and insoluble particulate matter as well as endotoxins were below general regulatory maximum limits on purified B-venom pharmacopuncture. And clinical safety test was also performed. Biochemical, hematological, histological toxicities were tested after vein or subcutaneous administration in rat for a short-term period. No death, clinical signs, ophthalmoscopic abnormalities and allergic reactions were found. In accordance of this result in animal study, human exposed to purified B-venom pharmacopuncture showed mild allergic reaction and more quick recovery compared to crude B-venom pharmacopuncture, demonstrating that the phospholipase A2 is a representative compound inducing hypersensitivity. In summary, B-venom pharmacopuncture causes allergic reaction, and this problem was resolved by removing the allergic substances and enriching the active compound melatonin. Our recent study provides useful information for safe and rational use of B-venom pharmacopuncture. In conclusion, we would like to just suggest that the improved strategies for industrializing of high-quality pharmacopuncture. Here, following several processes are essential for production of high-quality pharmacopuncture. First, certification of pharmacological raw materials. And second, standardization of manufacturing process. And third, quality control through efficacy, safety, and stability test. And finally, the first, industrializing, the first, establish the clinical data. Thank you for your attention.